Hi everyone, welcome back. We're going to continue our discussion on cells. And for this presentation, you are going to need the lecture guide that you should have gotten at CJ. You are going to need a blank sheet of paper or a piece of loose leaf to draw on. And you're going to need 4 different colors of markers or pen. So you can pause this and when you're ready, start again. So here we go. Cell organelles. All right. The last unit we talked about the 4 different types of organic molecules, the molecules of life, and you should know what they are. They are lipids, proteins, nucleic acids, which include both DNA and RNA, and carbohydrates. So, because these are the molecules of life, you would guess that these are the molecules that make up cells, which are the basic unit of life, and you are correct. Now, I don't know why biology books do that. They just talk about these molecules of life in one chapter and then you almost seem to forget about them in the next. So as I go through the parts of the cell, I would like you to draw along and color code it. So these are the colors I'm going to use. You can use the same colors or different colors. So let's go. Hopefully I'll remember what all my colors are. So let's look at the cell. All right, every cell you should already know has a plasma membrane. And the plasma membrane is a very, 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 very thin layer of, well, you can tell from my drawing, lipid. And this is a eukaryotic cell that I just did the outline of, and I will draw a little prokaryotic cell here, because that's part of your homework assignment, right? You should know the difference between a proc and eukaryotic cells. So all cells have a plasma membrane. So let's talk about the plasma membrane. So as I already said, the plasma membrane is a very, 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 very thin layer of lipids, or specifically fats, or specifically phospholipids. In fact, this layer, the plasma membrane is so thin, it's only 2 molecules thick. So the plasma membrane is so thin, it's only 2 lipid molecules thick. Now, besides being made up so again, it's only 2 lipid molecules thick, so we call this a phospholipid bilayer. But we'll talk a lot more about this in Chapter 7. Anyway, stuck in this lipid bilayer is little other little organic molecules. So you should be able to tell from my color what organic molecule this is. Do you need a review? Let's see if this works. Yeah. The purple represents protein. I chose purple for protein because your book always uses purple when it talks about proteins. And of course, not all proteins are really purple. In fact, probably not very many are. All right, so the plasma membrane of any cell is a very thin layer of lipids. Again, it's only 2 molecules thick, and it has these little proteins stuck all over it. And we could do the same thing here for a little bacteria cell. All right, so the word that you see associated with the plasma membrane is selectively permeable, or sometimes semi-permeable. So if we look at the little chart, the description I just gave it to you. It's a double layer of lipids, and it has protein molecules just floating around in the sea of lipids. The metaphor would be the guard gate of the cell. So what does a guard do? A guard says, halt, who goes there? And when something's trying to enter someone, and a guard will allow certain people to pass through the gate and reject others. And that's exactly what the plasma membrane does. It allows certain things to cross it and get in or out of the cell, but not others. So that's the plasma membrane. All right, I will just add why. Let's go back to green here. La -di -da -di -da. If we are looking at a bacteria cell, its membrane, it actually is convoluted and folded up in the middle here. And this is actually an extension of the plasma membrane, but it's sort of sealed shut, and this helps compartmentalize the cell. So anyway, so there we go. That's my bad drawing of the inner membrane, or the folded up membrane of a bacteria cell. All right. All cells, from our last lecture, besides having a plasma membrane, also have DNA. And I made the color of DNA blue because, again, that's what's color in your book. And you already know from prokaryotic cells, the DNA exists as one great big loop of DNA. We've already covered this, and little loops of DNA. Do you remember what those little loops of DNA are called? They are called plasmids. All right, but the DNA in a eukaryotic cell is found in the nucleus. Yeah, I'm just switching back and forth from these. So this is how I want you to picture the nucleus. This is my description. Picture a wiffle ball. You know what a wiffle ball is? Those plastic balls that kids play in, and what do they have in them? They have big holes in them. So I'm going to try to draw a two-dimensional drawing of a three-dimensional ball. And I don't know that I'm very good at it, but you know, I'm giving it a good old try. So picture a wiffle ball made out of lipid with a great big hairball in it. That sounds really appetizing too. All right, so what the heck are we looking at? So this is my wiffle ball made out of lipids, and where the holes are, those are supposed to be the three-dimensional holes. All right, and you'll notice, well, here, I'll tell you a thing. Now, I said picture the nucleus is a wiffle ball made out of lipids with the hairball inside. But of course, inside your nucleus is not hair. All right, these are long, 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 skinny molecules of DNA. So, the green double line is called the nuclear envelope. So I don't really care if you call this the nuclear membrane or the nuclear envelope, but sometimes it's referred to as an envelope because it's a double membrane. And each membrane is a lipid bilayer. So that would be 4 layers of lipids there. Whereas the plasma membrane is just a single membrane. So we have the nuclear membrane or the nuclear envelope. We have these big pores here. All right, I'm not drawing DNA here. I'm just showing you the big hole there. I'm just going down your lecture line. And then there's chromosomes in the middle. All right, so if this is, remember, you are made out of eukaryotic cells, and if this was one of your cells, let's say this was one of your skin cells, do you know how many molecules of DNA you would have in one of your skin cells? Yeah, a little review from honors biology. Do you remember? What if I asked you how many chromosomes you have per body cell? Would that be easier? Are you thinking 23, 26, 46, 23 pairs? All right. Let's do a little review. This is one thing you need to know for this next test. Every one molecule of DNA is the exact same thing as one chromosome. So we can say in every single one of your body cells you have 46 molecules of DNA or 46 chromosomes. I'm just writing out zome. I'm not writing out chromosome. All right, so you know what a chromosome is, hopefully. And the nucleolus is this sphere. It's sort of, you can see it under a microscope. It's a sphere inside the nucleus. This is this dark area. It consists of mainly of DNA, but some other stuff in there too. And the nucleolus is the site of ribosome synthesis. So this is where the ribosomes are made, inside the nucleolus, which is also on your lecture guide. All right, but so I gave you an overall description of the nucleus, right? It looks like a wiffle ball made out of lipids with a hairball inside, which is really made out of DNA. The metaphor would be the brain of the cell. That's a really good metaphor that you've probably heard before, because what does your brain do? Well, right now it's controlling that colored pen or pencil you have in your hand. It controls your body. Well, the nucleus is the brain of the cell. It controls all the cellular activities. And what else does your brain do? Well, it stores information for a test or a quiz. Well, that's exactly what the nucleus does. It stores information, but because it's part of a living organism, it's storing genetic information. So the two functions of the nucleus as a whole, it again controls all the cellular activities, all the goings on in the cell, and it stores the genetic information. All right, I think I've covered everything on nucleus, nuclear envelope, nuclear pore, chromosome, and nucleolus. Okay, now we're down to ribosomes. Ribosomes are little spheres of RNA, all right, and you find them, some of them floating around in the cytoplasm. And remember, and I'll put some of these in a prokaryotic cell too, and I just gave it away. Again, these are little spheres of RNA, so that's why they're blue, because they're nucleic acid, just like DNA. So remember, so we've drawn now the universal components of cells, right? Both prokaryotic cells and eukaryotic cells have a plasma membrane. So we got that. All living cells have DNA. We drew DNA into both. All have ribosomes and all have cytoplasm. And hopefully you remember the function of the cytoplasm. The function of the cytoplasm, the function of the ribosomes. The function of the ribosomes are to make proteins. So here I'm squirting a little chain of amino acids out of this ribosome, and here we're squirting a little chain of amino acids out of this ribosomes. And this gives you a little appreciation for scale, because when we covered proteins, proteins were huge biological molecules. Yeah, well, they are. But compared to a cell, right, the protein molecules are quite small. All right, so that's ribosomes. So now we're going to go to the ER, which are the endoplasmic reticuli. All right, so here I'm going to draw the smooth ER, and the smooth ER you can see is made out of lipids, and it's a branch of interconnecting canals coming right off of the nuclear membrane or the nuclear envelope. So the smooth ER and when I draw the rough ER, they both are continuous with the nuclear membrane. So now I have to find my pointer and draw a rough ER. Oh, let's draw a rough ER right here. So the description of the ERs, an endoplasmic reticulum, one endoplasmic reticulum is a series of interconnecting membrane canals. Again, I'm trying to draw in 2 dimensions. What's 3 dimensions? Now, what's the difference between rough and smooth? Well, I have to draw one. All right, guess which ER is the rough one, the one on the right or the one on the left? And if you're saying the one on the right, you would be right. And hopefully you can tell from my lovely drawing, what I'm drawing on the outside of the rough ER are ribosomes. So the smooth ER has no ribosomes attached to the outside of it. The rough ER, that's sort of easy to remember, rough ribosomes. All right, so that's the description of the smooth ER, series of interconnecting membrane canals. Now I'll go over the function. The function of the smooth ER is lipid synthesis. Do you know that word, synthesis? Some students do and some students don't. Okay, synthesize, to synthesize something means to make something. So the site of the function of the ribosomes was protein synthesis, and they were the protein factories, and now the function of the smooth ER is lipid synthesis. So it makes lipids. All right, so the function of the smooth ER is to make lipids. Another function of the smooth ER, if let's say it's in a gonad, so another function of the smooth ER, if this body cell is located in the gonad, would be to make sex hormones. So the side of a smooth ER could be to make lipids, and the side of the smooth ER is to make sex hormones. Now, right now, what I'm about to tell you, this is the difference between memorizing biology and understanding biology. If you really understand the material from Unit 1, you should know why, if a function of the smooth ER is lipid synthesis, why it would make 100 absolute total sense that it would also make sex hormones. Can you think about it? All right, I think you're doing this at home, so I can't have you talk to your neighbor. So write down your reason. Why does it make sense? Right now, go. All right, hopefully you remembered there's 2 types of lipids, right? Fats and cholesterol. All right, and the plasma membrane was made out of the type of lipid called fats, and this smooth ER makes both fats and cholesterol. And remember, cholesterol is the steroid mama. Your body takes cholesterol and turns it into the sex hormones. So this should make perfect sense to you. If the smooth ER is making cholesterol anyway, what's the big deal to modify a cholesterol molecule and make a sex hormone out of it? All right, got it? And I don't think I have a metaphor for the smooth ER. So let's go to the rough ER. The rough ER has the ribosomes on it. Well, the function of the rough ER is to transport proteins, which makes sense because it has ribosomes on the outside of it, and these ribosomes are squirting proteins into the middle of the rough ER. Now, some proteins the cell keeps for itself, and some protein the cell exports. So hopefully you would figure this out for yourself. Here's a ribosome hanging around the cytoplasm, and the protein it squirts out, it's going to keep for itself. This might be an enzyme it needs in the cytoplasm. But these proteins, these are bound for export. So that is the function of the smooth ER. I'm sorry, the rough ER. It's going to transport proteins that are going to end up on the outside of the cell. Now, why would a cell do that? Why would it make proteins and not keep it for itself? Well, let's say this cell happens to be in your pancreas. And what does your pancreas do? It makes a very important protein hormone called insulin. So when you get this spike of sugar because you just decided to eat a nice, wonderful chocolate chip oatmeal cookie, and you eat this chocolate chip oatmeal cookie and you get a blood glucose spike. Well, what happens? Your body, more specifically your pancreas, has to produce insulin. And it doesn't keep it for itself. It needs to make insulin and then transport it out into the blood. So if this is a pancreatic cell, it could be following the instructions from the nucleus and making some protein, which will eventually end up outside the cell into the bloodstream. So let's go on to our next organelle. Well, I see after Golgi apparatus is the word vesicle. So this is what happens is, remember, this is supposed to be three-dimensional. So pieces, where is my pointer? There it is. Pieces of the rough ER will bubble off. That's supposed to be an arrow. And this is a three-dimensional membrane bubble. And this three-dimensional membrane bubble is a vesicle. So these are bubbling off here all the time. So the metaphor for these vesicles, well, first, the description of these vesicles would be a three-dimensional membrane bubble. And the metaphor is a delivery truck. So what happens? These vesicles fuse together and they make the Golgi body. And the Golgi body is basically this stack of membrane pancakes or membrane pita breads or whatever. So they come in and these bubbles fuse together and make these stacks of membrane pancakes or membrane pita breads. Now, what was inside the rough ER? Well, there were proteins inside the rough ER. So, as you would guess, there's proteins inside the vesicles. So in this story that I'm telling right now, these vesicles carry a pre-insulin molecule, and they're going to fuse together. And so in the Golgi body here, this whole stack is the Golgi body, there are proteins in here. So the proteins were squirted from these ribosomes into the rough ER. Then the rough ER bubbles off, and you have the pre-insulin in here. Now they're fusing together, and now we have pre-insulin molecules for the sake of the story in here. Well, my metaphor for the Golgi apparatus is like an Amazon warehouse. All right, so, let's see, Cameron's buying a new pair of shoes, and Teresa's buying a new book, and I don't know who's buying what. I don't know. Emily's buying something else. All right, maybe Emily's buying shoes too. And so Amazon has all these orders, and it has to package these orders so the right shoes and the right textbooks get to the right people, because we don't want, Emily's shoes being delivered to Cameron. So the metaphor for the Golgi body is Amazon.com warehouse. And the function is it's going to package these proteins. Now, if you're really in Amazon.com, you're going to package the shoes or the textbooks with cardboard and then label it, but your Golgi body does not have any of that stuff. So what the heck does it label the proteins with? Well, I don't know if you can remember what this color is. What I'm adding are little tiny pieces of carbohydrate. So the Golgi body is adding carbs for the label. So once they're all labeled, they are ready to be shipped out. So what's going to happen is they're going to get in another delivery truck. So we're getting another vesicle here. So it bubbles off, and now what we have inside these vesicles are the mature insulin molecules that I'm not drawing, and this is supposed to be arrows. And this vesicle fuses with the plasma membrane, and then when it's fused, right, it dumps its contents into, let's say, the blood. So now I'm going to add my proteins. La-di-da in color. So these vesicles, the delivery truck, they had the protein particles. I did not mean to draw that line. I wonder if I can do undo. Nah, I'm not that talented, sorry. So I'm just so not talented here. So these I'm drawing proteins in the vesicles that are insulin, and then they get dumped into the blood. All right, so there we have it. What else do we want to say? We haven't gone over lysosome yet. So what a lysosome is a specialized vesicle. So it is also a membrane bubble, and might have bubbled off the Golgi body. And this lysosome will contain protein, but these proteins are all digestive enzymes. Yes, indeed. So since a lysosome is a specialized vesicle with digestive enzymes, you could guess what its function is. It would be digestion. So that's my overview of the cell so far. It looks like I left mitochondria out, but I guess I'll just talk about that in person. So go ahead and listen to the second lecture on cell organelles and take some additional notes. And we'll please know this when you come. Everything I covered in this lecture, please know when you come to class on Tuesday so we can expand upon it. I will see you then. Bye.